All right, thank you everybody for attending the second chair, the biotic stresses under climate change. As you all know and you're all aware, climate change and its drastic temperature swings that we're seeing, and as far as precipitation, is causing major problems as far as barley production, especially in malting barley quality. But when we always associate that with drought and high temperature, there's also the fact that in certain areas we have drought and high temperatures, but in others we're seeing higher moisture, higher temperatures, and we're seeing the emergence of new diseases on barley that were not traditionally there. And that's something we've seen in the Pacific Northwest with stem rust and other places around the world. And so one of the tools that we have to be able to breed for wide adaptability, which is very important now in these situations, is the wild barley germplasm pool. And so our keynote speaker today, Professor Brian Stephenson, will be talking about his work in developing and unlocking this diversity in the wild barley germplasm pool. And a quick introduction, Brian has been huge in the barley pathology over many years, and he started that at North Dakota State University and then moved on to Minnesota State University, where he is the Lieberman Okinow Endowed Professor of Cereal Genetic Resources. And he's, with further ado, Brian Stephenson.
Thank you very much for the introduction, Robert. And I want to first off thank the organizing committee for inviting me to this symposium. It's a great honor. And I'm actually looking forward to speaking with all of you in person. And I think the best scientific talks come after a few beers when we're meeting informally. So I'm really looking forward to that. Well, before I left for Latvia, I discovered that there were many Latvians who were coming to my hometown in Minnesota. And there is a festival that occurs every few years, the Latvian Song and Dance Festival. So there were quite a few people coming into town. And I found out that we had a very high-level person come to visit, and that was your Prime Minister. So we would like to reciprocate for this 13th International Barley Genetics Symposium, but President Biden sends his regrets. And all we have is Kevin Smith and myself to represent the United States. So I hope that's okay. So for outline of my talk, I want to just briefly speak about biotic stress under climate change, and then talk a little bit about the genetic characterization of the wild barley diversity collection for disease resistance, which we've been working on for a time. And then I'll move to speak about the establishment of the International Wild Barley Sequencing Consortium and just the very first tests that we've done on genome-wide association studies with those data. And then finally, allude to and talk a little bit about the potential of the pan-genome in terms of gene discovery, especially for disease resistance genes. And Martin Mosher set up that talk very well, and I appreciate his talk earlier. So it, of course, is undeniable that global climate change is occurring, and we need only look at the weather from day to day to see all the catastrophic heat and drought losses, severe storms, flooding, and what have you, to really know that the global climate has changed in the world. And probably the most key factor of global climate change, of course, is the temperature. And the recent temperatures that we had are now the hottest that we've had in the past 2,000 plus years. And you only need to look at some of the data provided by NASA to see how the global temperature is changing over time in our area. And that's not boding very well for barley, which of course is a cool season crop. And in a study that was done by Shi et al. A few years back, they looked at various climate models for the past 90 or the future 90 years and found that the average barley yield reductions can range anywhere from about 3 to 17%. And this graphic in this slide shows that most extreme model where there's a predicted loss of 17%. These workers also looked at the effect of on beer supply and price, and that they found under the most extreme model, beer consumption could decrease by 16%, and the price could double. What a terrible thing. So instead of seeing all these happy people enjoying a nice couple of cold beers out at a pub, we might see something a little bit more depressing, like this gentleman drinking alone. But it's easy to understand how climate change can affect plant disease, and we need only go to that basic principle of introductory plant pathology of the disease triangle to know that changes in climate can definitely cause a more favorable environment for disease. I mean, you could just say that more wetness that favors fungal infections can be something that will increase diseases, and also perhaps temperature. We can also imagine there would be greater virulence and fecundity in the pathogen, again, probably due to temperature. And we could also imagine greater susceptibility in the host, for example, temperature-sensitive genes. And I wanted to just speak briefly to one example of that. And that's work that was done on stem rust of barley with actually our chairperson, Robert Brueggemann, and several others. But this stem rust pathogen is a really devastating pathogen that could destroy crops over a wide area in just a short bit of time. Probably the most prone areas in the world are North America, Eastern Australia, and East Africa. The one that we're concerned about most that you've probably heard related to wheat is UG99, or as they call it, race TTKSK. That was found to be virulent on about 80% of the world's wheats, but in work that we had done recently, we found that the pathogen is virulent on 95% of the world's barley cultivars worldwide. And it consists of about a dozen variants that are spread across East Africa, South Africa, and into the Middle East. But in work that we've done in terms of screening barley for resistance, we have found this RPG4, RPG5 resistance gene complex to be really the best resistance genes that we have against this pathogen and other pathogens as well, other pathotypes as well. The nice thing about this gene complex is that it can be really readily recognized by low infection types at low temperature. And the low infection types I'm speaking of are the 0 fleck or 0 semicolon, as our notation goes, or 0 fleck 1, which shows a little sporulation of the pathogen. It's a very distinct phenotype, really easy to pick up. However, if you screen this material at high temperature, so instead of 19 degrees centigrade, you screen it at 27 degrees centigrade, you find that the resistance really starts to erode dramatically. So from like a 0-0 fleck reaction, as you see in this left part of the slide, you see more of the 3-2 type of reaction. So a really strong erosion of the effectiveness of the gene. Now that's done in the controlled conditions of the growth chamber. What happens to this gene in the field? Well, in the field, genotypes with this gene complex can actually suffer up to 40% severities when you have warm summers. And sometimes I thought initially that we had the wrong seed and we had a mix-up, but we found that this is really how this gene responds during very warm summers. Now, in terms of looking at genetic diversity, I like to show this slide showing the different grains, fruits, and vegetables with their myriad of sizes, shapes, and colors to hit home to students that there's a virtual treasure trove of genetic diversity that exists both in wild species and land races. And of course, this is only the phenotypic diversity that we're seeing. We know that underlying that there's a tremendous more amount of genotypic diversity. So in our focus at the University of Minnesota, we're really looking at enhancing disease resistance in small grain cereal crops with genes derived from the wild relatives. And there's been a number of really wonderful talks this morning who have set up this topic. But what we're working on really is the primary gene pool, barley's progenitor, and that's Hordeum vulgare subspecies spontaneum, or I'll just say spontaneum for short. So just a little background about spontaneum. As you know, it's native to the Fertile Crescent area, and it's really one of the most cosmopolitan species that you find across that region. I've traveled a lot in the Middle East, and you can look around the landscape and you'll find wild barley and also wild oat growing all over the place. And this is kind of in contrast to a wild wheat that I work on, wild emmer, that found in more specific sites where there's basalt rock formations. And we also are fortunate that wild barley can hybridize readily with cultivated barley. There's no barrier, so we can transfer those genes quite easily. And the other thing is we know that there's a tremendous amount of diversity in the wild spontaneums. In whatever trait you happen to look at, you'll find variation. So in order to kick off this work, we wanted to get a good collection of germplasm, and so for this germplasm collection, we naturally went to the International Center of ICARDA, which was based in Aleppo, Syria at the time, and the curator at that time was Jan Valkoen. And so Jan helped put together an ecogeographically diverse collection of spontaneum for us that, of course, is very heavily sampled where the high populations are in the Fertile Crescent area. Some 77% of the accessions are from there. Also, there's quite good sampling in Central Asia, a little bit in North Africa and the Trans-Caucasus region. So this slide of the world here shows where those accessions are from and some information about the subpopulations that we have found in the subspecies. Now, of course, since I'm a plant pathologist, I'm specializing in diseases, and we thought that we would run this collection across the myriad of different pathogens that we work with. So we've tested a number of races of each of these diseases, and then I've had other cooperators who have helped test the materials well for the disease reaction. And basically for our objectives is really, as far as an applied objective, is to identify new effective resistance genes and then to be able to transfer them into the breeding program. So we've kind of undertaking a pre-breeding type of approach. And then the other thing we're interested in is to characterize the genetic architecture of disease resistance in wild barley. And finally, if GMO type of barleys are allowed, we think that one really quick way of achieving durable resistance is to clone multiple resistance genes, assemble them in cassettes, and introduce them into barley by transgenesis. And I think I was really influenced by the success that we've been working with in terms of Mick Ailiff's studies. So he's been working on putting together these gene cassettes, and he put one cassette together that has 5 resistance genes. One of them is a multi-pathogen resistance gene, and that's the SR55, LR67, and YR46 gene from cultivated wheat. But he also put 4 other resistance genes that were cloned out of various wild wheat relatives into this 5-gene cassette. And we were privileged to be able to evaluate these materials in our nurseries, and I have to say I haven't seen anything quite as impressive as how resistant these materials are. So we're very optimistic that we might be able to control the wheat rust trifecta, as I call it, of stem, leaf, and stripe rust with cassettes of this sort. So in our beginning work, we were looking, of course, at the genetic architecture of disease resistance in wild barley. So we were using the genome-wide association study route. And we've done this for a number of different resistance genes and come up with some really nice patterns in terms of peaks where resistance genes lie. But one of the things that we quickly found, and I know many of you are aware of, if you have a allele at very low frequency, it will go undetected in the GWAS plots. And so we're interested in many of those very, very rare alleles. It seems like inevitably the highest value genes are the ones that are extremely rare in this collection. And as an example, we discovered another stem rust resistance gene called RPG5. It apparently wasn't really readily detected in the GWAS plots, but we had to go through the normal biparental mapping, and we're able to map this new gene to the long arm of chromosome 3H. And then my colleague at Oklahoma State, Dolores Morhenweg, had screened the collection, and she identified two new genes for resistance to green bug. And again, they were in very, very low frequency, and they were mapped again by a classical biparental approach. Now, in working with Branda Wolf on some of the materials that he was interested in with wild wheats, he was coming up with various techniques to try to clone genes more efficiently. And he came up with sort of a genome representation reduction method of combining association genetics with resistance gene enrichment sequencing. And it was purported to be a really great method for being able to detect not just one, but potentially the entire complement of the NLR type resistance genes that you might find in a species. And for a proof of concept, he used the D-genome donor of wheat, Aegilops tauschii. And Branda was at the John Innes Center initially, and now he's in Saudi Arabia at KAUST. But just to show you some results from Branda's study, we knew that one of the few genes that tauschii donated to wheat breeding was SR33, and that peak came up very clearly in this RenSeq plots. And then the other two new genes that they discovered were SR45 and 46, and then also one TA1662. And they were able to clone these genes in fairly fast fashion. Now, we thought that this would be a great method for us to use in our wild barley as well, and so we applied this to the wild barley diversity collection, and we screened that collection against 10 different races of the leaf rust pathogen Puccinia hortii. The more different pathotypes you can use in this screening, the much better that you can parse out individual genes. So we hope that with our panel of 10 isolates, we could do this quite readily. And just to give you a very quick summary of the results from this work, we found 144 sequences that were associated with the reactions to the 10 different races of the leaf rust pathogen. And one of the most remarkable Manhattan plots that we had seen is shown in the upper right. We had this really clear definitive peak that was found to be on chromosome 2H. And later on, we found out that this gene was actually RPH15, a gene that we had described from wild barley about 15 years previous. So it's a really great gene. I have a very big collection of leaf rust isolates. There's only one that I know of that can take out this gene. So it's a really great high-value gene. So we embarked on trying to start cloning this gene, and fortunately, I was in discussions with my colleague in Australia, Robert Park and Peter Drakatos, and we'd come to find out that we hit the same gene that they started cloning, and so we sort of pooled our resources together, and eventually RPH15 was cloned with the group from Australia. Now, 4 other RPH genes were cloned in this work, but we have not yet validated them functionally with transformance. So before I would recommend anybody embarking on AgRenSeq, I would like to just point out that there are some shortcomings that we encountered. One is that the distribution of some of these candidate genes did not follow a nice, clear dichotomous pattern in that they're present in the resistant and absent in the susceptible ones. And really, instead, we found the genetic basis to be a lot more complex and involves specific alleles that differed in their SNPs. The other problem was that these contigs contain really very small portions of the regulatory sequences, so we're having to do a fair bit of genome walking to capture the region. And we knew from the outset, of course, that the way the baits that are used for this process are using the NLR type of resistance genes. That would be the only type of genes you would identify, and you'd miss some of the other classes of R genes. So the next sort of thing would be to try to get some sequence data for these materials, and that was part of the genesis for establishing the International Wild Barley Sequencing Consortium. So this was sort of thought up during the COVID period, and I was thinking, how can we sort of create genomic, genetic resource for the barley community, and also maybe help us capture some of these genes. And so that sort of began the campaign to write to many of you, and you were kind enough to contribute to this project. And I'm here to tell you, like, what happened to your money that you donated? And but anyway, we raised about 150,000, so it was enough to cover the sequencing costs that were done at IPK. And it's really great that we had such a wonderful response from everybody. So we had like 61 researchers from 35 different institutions in 14 different countries that participated in this consortium. And just to give you a few short summary statistics, we have sequenced on 285 of the 314 total wild barley sessions. We eliminated some because they were very, very closely related and they weren't going to yield a lot of useful information. So we have 285. We have a lot of data that was generated, somewhere around 10 terabytes, and we achieved about 10x coverage on these materials. Muru, who works with Martin, just informed me that he just got in a lot of this data. It was very new, and he called about 114 million SNPs out of this material. Another worker is helping us on the GWAS, and that is Lior Glick at Tel Aviv University, and he gave us a statistic of K-mers, 31 count K-mers, of being about 2 times 10 to the 9th in terms of the number on average. Now, people are probably wondering, like, when is the data going to be released? When are we going to see the initial publication? And in speaking with Martin, we think somewhere around the September date is when we'll have the publication ready. And we hope to release the data as soon as we can, maybe sooner than September if we can all get in agreement. Now, just to update you on the infamous collaborative agreement, it looked like it was going to get finished until we had certain lawyers get involved in certain countries, and so it's still at a little bit of a stalemate, but I think we're almost through, and it may require that you have to sign once more. I apologize for that, but it will be done, and then we can go on with the evaluation of the data. So I apologize for that, but that's what happens when you get lawyers involved. Now, I want to make a little pitch for the wild barley diversity collection. So many of you are probably, plant breeders and say, I don't want to mess with that unadapted junk, but it really has a great potential for enhancing barley breeding. And I've done now with my colleagues about 57 different disease data sets. The pathogens run from 1 to 40 different races. The one, if you want to find out who did the one with 40, it wasn't me. It was my colleague in the Czech Republic, Antonin Dreisaitl, who screened the entire collection with 40 different isolates of mildew. So that is really a wonderfully rich data set. But we've also done, in addition, many agromorphological traits, microelement content. The only abiotic stress tolerance we've done is to cold hardiness, and believe it or not, this Mediterranean-based species actually is hardy enough to survive Minnesota winters, and we're using that in the breeding program. We also have some insect resistance that was found, as I said, by Dolores Moore-Henwig. We've done nutritional components and also looked at malt quality with regards to beta-glucan. So my pitch here is that if you have any interest in looking at allelic diversity, be happy to have you join in and tackle some of these traits, because it's far more than what my project can do. So we would be really happy if other people took on the gauntlet to get some of this data analyzed. Okay. So just to end up on the last bit that I want to show, this is just very recent data from Muru and also Lior Glick, and this shows the first GWAS that we've done on SNP-based GWAS and also K-mer-based GWAS. And what I want to point out very briefly is that we know that it has identified RPG405 because we've done the analyses there. So that's indicated on the top graph with the star. And there are other stem rust genes up to 7, and we've positioned those in the blue, but all those other red points across there represent potential novel alleles for stem rust resistance. So we're just scratching the surface really in terms of the allelic diversity we can have to this one disease with just one race. And you can see at the bottom of the screen, the K-MER analysis revealed fewer peaks, and that's to be expected, I guess, when you're dealing with 31 MERS. So Martin talked about the current pangenome resources, so I won't speak much to that, but upcoming for the pangenome is these additional ones where we'll have 17 wild barleys and 58 cultivated barleys. And future research is probably going to need to require some transcriptome data be analyzed and maybe eventually pack biosequencing for certain individual target accessions you might be interested in. And as I said, we would like to have additional phenotyping done, and the seed is available at USDA, and they'll send it to you free of charge with a phytosanitary. So just a real quick summary, this wild barley diversity collection is really a treasure trove of unique alleles for disease resistance. I'm sure it'll be very valuable for other alleles as well. Our AgRen-seq program, while moderately successful, is really going to require whole genome sequencing and other analyses. And I think that eventually, if we clone enough genes, we can use that cassette sort of technique for making sure that barley is durably resistant. Lots of people contributed to this. I want to just show the individuals who contributed a lot to this project and also the funding sources. And again, I want to thank all the people who generously donated to the sequencing consortium, and we hope to get that data to you very soon. I want to end with a very short tribute of a very significant Latvian, and that's Andy Kleinhoffs, preeminent barley geneticist. He was born here in Latvia, came to the US when he was 8 years old, and he was very instrumental in a lot of the first resistance gene cloning of rust resistance genes with Robert Bruggeman in my career. And he really wanted to come to this meeting when it was announced 2 years ago, but sadly he died on May 9th. So he was a big man, but even still he was larger than life with that. And I just wanted to acknowledge Andy and his contributions. So thank you very much for your attention. And if we have any time, we'll try to answer questions, but thank you.
Thanks, Bob, for the introduction. As you said, my name is Carl Efforts. I work at Washington State University, and today I'm going to be talking about one of those R genes that Brian said the AgRen-seq could not capture, which is RPT5, a net blotch resistance gene in barley. And so I just wanted to give a quick introduction to net blotch, even though Brian talked about it briefly. It is a necrotrophic fungal pathogen with typical yield losses of 10 to 40%, although if you have a susceptible variety in conducive environment, you can see total loss. There's also a very concerning thing, especially for American farmers, that you get a reduction in seed quality. So if you're trying to grow malt barley, you need a big plump seed, and when net blotch infects the barley, it can be shrinking the seed kernel, and so that would get a reduction at the elevator. There are two distinct forms of net blotch: spot form net blotch, which you can see forms spot-like lesions, and then net form net blotch, which is the pathogen that I study. So this talk's going to focus on net form net blotch. And I wanted to get in briefly into the genetic interactions between pathogens and their hosts, since that's kind of what this talk centers on. And so with biotrophic pathogens, such as rust, you see this gene-for-gene interaction. And that's where you have a dominant avirulence protein from the pathogen and a dominant resistance protein from the host. And this results in a very controlled programmed cell death. And since biotrophic pathogens require living tissue to proliferate, if the host kills off, a very small number of cells, it can stop the pathogen from proliferating, and resistance is the result. Now, with necrotrophic pathogens, almost the exact opposite occurs, and we call this the inverse gene-for-gene model. And that's where a dominant necrotrophic effector is recognized by a dominant host susceptibility protein. Again, the same result is programmed cell death, yet since necrotrophic pathogens feed on dead or dying tissue, they can proliferate and cause tons of disease like you see here on the right. And I briefly wanted to go into the research that's been done previously on the genetic resistance to Pyronophora teres form teres or net blotch. And so this is a figure from a book chapter that we published last year. And this was put together by my lab mate, Dr. Sean Clare. And what it's showing is the 7 pairs of barley chromosomes. And On the left, visualized, is the SNP density from the 50K iSelect chip. And on the right is a heat map of how many times these loci have been implicated in Pyronoptera teres resistance. And you can see on that arm of chromosome 6H is that very dark green patch, and this is the locus I'll be talking about today. This is the RPT5 SPT1 locus, and RPT5 is a robust source of resistance against net blotch. It's been reported in over 20 publications. And it also is isolate-specific susceptibility, or SPT1, that our group has maps to this exact same locus. And moreover, all of the identified PTT effectors have been found to genetically interact with this locus. So it seems like the pathogen is keying in very specifically on this area, and there's something interesting happening with how the plant is maintaining it, which I'll get into a little bit later. So we wanted to dive into this, and this work was done by my predecessor, Vaidehi Kaladia, and she QTL mapped RPT5 using a CI5791 by Tefang recombinant inbred population. CI5791 is a Ethiopian landrace of barley that is highly resistant to almost every known isolate of Pyronoptera teres form teres. So she phenotyped with 9 diverse PTT isolates ranging from Denmark, Japan, Brazil, and the US, so a pretty good global collection. And then genotyped with the 9K iSelect chip and identified 2 major QTL, this one on 3H, and then this sharp peak here on 6H, which she found to be effective against all 9 isolates tested, and it was contributed by line CI5791. So this is where I joined the project, and we wanted to high-resolution map RPT5. So we did this. I think the original range was something like 50 megabases and contained 300 and some genes, so obviously far too early to start gene validation. And so we saturated this region with SNP markers, and then through using a CI5791 by Tefang F2 population, we were able to do high-resolution mapping. And we got it down to, this was when the original draft assembly of Morix was out, or the original first assembly, and so it was approximately 3 megabases on that assembly. Looking a little bit more closely at this locus, and this is just from one of the pangenome lines, we see that in most pangenome lines it's actually much smaller, and it's in a very, very gene-poor region. It's near the centromere, and so it's very repeat-rich and only has a handful of genes. And we identified 3 genes within there that had homology to known resistance genes. And at the same time, another member of our lab, Dr. John Richards, he was fine mapping the SPT1, that isolate-specific susceptibility. He did this with a Rika bicombar F2 population, and those 2 have reciprocal virulence with PTT isolates 15A and 6A. And so he was able to delimit this region, and when we looked at our results side by side, they're the exact same region. And so we definitely think that there are potentially multiple genes or possibly just different alleles within this region that are contributing to either a resistant or a susceptible outcome. And we do have a candidate gene identified with this that I'll touch on just very briefly towards the end of my talk. Another interesting phenomena that we observed was this double recombination that happened in very, very specific locations during the mapping. And so in between 2 of our markers, I think it was about a 50 kilobase or 100 kilobase, I can't remember exactly the distance, we consistently saw recombination there, and we consistently saw the recombination correlate with disease outcome. And so I want to do a lot more work on this, but I just wanted to touch on it since we have all of these pressures pushing in on this locus from the pathogen. And so when we looked at those 3 genes, we looked at the alleles present, and then we screened a selection of the World Barley Core mini-core collection. And what we found is, I think we screened with 4 PTT isolates, and we found that around 30 of the lines were highly resistant to all 4. And when we looked at the alleles for our candidate RPT5, we found that almost all of them contained or harbored that CI5791-like resistance. And then down here at the bottom of the slide, I just have a protein model of what this gene looks like. So it's somewhat a receptor-like protein. Some transmembrane domain prediction softwares predict it there, some do not. And so I'll get to that a little bit later, but we wanted to tag this protein with YFP and see where it actually goes. And it's highly variable between parent lines. You can see this is an amino acid alignment, so it's got some very, very strong diversity. We call this the hypervariable neck. But lots and lots of differences, and then also differences to the leucine-rich repeat binding pocket. And so with that in hand and the correlation with highly resistant lines, we decided to go forward with complementation. And we did not have the capability to do this in-house, but so we did this with a collaboration with Dr. Matt Muscal from the Sainsbury lab. And we cloned the full-length RPT5 into this PBRAC202 vector. At that time, this was back in 2017 or 2018, we did not actually have the full promoter sequence or full terminator sequence from RPT5 from our resistant parent CI5791. So we elected to put in an MLA6 promoter and terminator, which will become relevant a little bit later on. And all the transformation work was done at the Sainsbury lab, and then the seed was sent back to us at Washington State University for phenotyping. And so our phenotyping results, we initially screened with PTT isolate 6A, and we noticed a dramatic shift towards resistance. So you can see on the far left there, highly resistant line CI5791 is about a 1 on our 1 to 10 scoring scale. And the transgenic lines are there in the middle, and they averaged about a 4. So about half as bad as the parental line Golden Promise that single gene was transformed into. We see a pretty strong reduction in lesion size and then a huge reduction in chlorosis. And since this is a very broad resistance gene in the resistant parent CI5791, I wanted to take a look at a couple other isolates. And so we did it with the isolate PTT0-1, which is highly virulent on 6A. Highly virulent on Golden Promise. And you can see that while they still do have pretty strong disease, the transgenics do have a good shift towards resistance as compared to line CI5791, which shows that nice, just little flecking. And then additionally, we looked at a moderately virulent isolate, PTT15A. And so given the fact that we weren't actually able to clone in the full-length promoter from CI5791, we wanted to look if perhaps expression data was giving us those differences in phenotype, or if it just happens to be other genes or other resistances present in the genome. We did see that expression of RPT5 did correlate with disease outcome. You can see CI5791 and our transgenic there, which is labeled HVT, they have somewhat comparable expression levels before the pathogen has been inoculated. However, the second bar in is CI5791 48 hours after pathogen inoculation. And you can see it's got almost a 4-fold induction, whereas the transgenic one, since it has that MLA6 promoter, does not have that induction. And then just briefly, I talked about how we had a candidate gene for our SPT1, and we found this through some RNA-seq data and saw that it was really highly upregulated in susceptible interaction. So we have qPCR data shown here from Golden Promise, but with several other cultivars and isolates where they have a susceptible outcome, you see this massive upregulation. And this gene is a receptor-like kinase or encodes a receptor-like kinase that is within our locus, and so we think it may also be involved in that dichotomy between if it's a susceptible or a resistant outcome. I just wanted to quickly touch on some of the functional characterization that we've done. So, like I said, it has, in some alleles, a predicted transmembrane domain. Some programs predict it, some don't. So we did tag it with RFP and agro-infiltrate that into Nicotiana tabacum leaves. And we did see that it does localize to the cell membrane. And it might be hard to see on these slides, but it does seem to form these aggregates on the membrane that we think might be lipid rafts. And that kind of has importance because we've found, very recently, we've found a yeast-2 hybrid interaction between a couple lipid-modifying enzymes and RPT5. And then also through yeast-2 hybrid, we've found a couple interesting interactions with the PR proteins or pathogenesis-related proteins. These are small apoplastic proteins that are involved with tons of different pathogen invasions. And we found that RPT5 from line CI5791 interacts with a PR1-like protein. And then we found a virulence effector that targets this same locus, interacts with two different PR1 proteins. So that's kind of where my next work is going to go, because I kind of want to explore what are the pieces of this proposed model that we're missing, because we don't really have a lot. But we are hypothesizing that the extracellular portion of RPT5 is interacting either with fungal PAMPs or interacting with or monitoring the interaction between fungal effectors and PR proteins. There's been some really interesting work recently in Parastagnosporum that has shown a specific interaction between a fungal effector and a PR gene is noticed by a resistance receptor, and then it's able to transduce a signal and upregulate resistance response genes. So just quickly wanted to go over the impact and the future work. Through this work, have cloned and validated RPT5. It is the first resistance gene cloned in the barley PTT pathosystem. It is the most robust source of resistance that we know about in the pathosystem. And the pathogen appears to be keying in on this locus and causing some very interesting genome dynamics to occur. Functional characterization, it appears that possibly the PTT effectors are targeting PR proteins, and maybe this is what RPT5 is monitoring, but there are still many pieces to uncover. And then future work, I do want to look into that SPT1 candidate gene in more detail and determine the other genes involved in this resistance signaling. So with that, I'd like to just acknowledge everybody in my lab, the barley breeding and molecular genetics group at Washington State University, especially Bob and my lab mate, Dr. Sean Clare. And also like to acknowledge Dr. Muscow at the Sainsbury Lab, Dr. Tim Friesen at the USDA-ARS, and then the WSU MPS Student Travel Grant, because that's the money that got me here. So, and also my funding agencies, NSF and USDA. With that, I will open it up for questions if we have time. Sorry, I don't know if everyone heard this question, but there are Ethiopian isolates that are able to circumvent this. And we do not have clearance at our lab to have any of those isolates, although those would go through Dr. Tim Friesen. One thing we want to do is challenge these with, what is it, SM, one of those Moroccan, there's an isolate from Morocco that's broken RPT5. And so we want to take our transgenic and test them with that and see if it goes away. And so that'll definitely be a next step.
Yes, just out of curiosity, I mean, I hypothesize that your differences in gene expression in your transgenic lines might be due to different copy numbers. Have you actually checked into this, or are you planning to?
They're all single copy.
Hello, thank you so much. So yes, I'm Molly. Thanks for introducing me, and I work with Matt Moscow. And I feel very fortunate to be here today, so thank you for your attention. I'll jump right into the plant immune system. And so it's generally regarded as a two-tiered innate immune system. And this gives plants the ability to respond to pathogens. So in the extracellular space of the cell, pattern recognition receptors respond to patterns either from modified self signals or from pathogens. And let's see. These include both receptor proteins, as was mentioned as the candidate gene by Carl in the previous talk, and receptor kinases. And the difference between these is just that the receptor kinases have an intracellular kinase domain, and this is integral to transducing the signal inside of the cell. So recognition also takes place inside of the cell, and this is primarily mediated by NORs, and these either indirectly or directly respond to effectors from microbes. So in this talk, I'll be talking about a receptor kinase. Specifically, this receptor kinase is Puccinia striformis receptor kinase 1, and we call it PER1. In this talk, I'll be discussing how it was identified from a locus on the long arm of chromosome 4H called RPS8, And I'll be talking about the interesting finding that another gene in this locus, XO70FX12, is also required for its function. A paper is about to be available online in 3 days, and this was led by a previous PhD student, Sam Holton, in the group. And so we're very excited about this paper, and I'll be covering a lot of data from this. To begin, the Mosca group identified 3 resistance loci in barley that are responsible for maintaining host specificity against the wheat stripe rust pathogen. And these are RPS 6, 7, and 8. I'll be talking about RPS 8. And you can see that this locus in isolation is enough to confer resistance in barley against wheat stripe rust. And you can see that because there are no pustules forming on the leaf surface. We mapped RPS8 to a 936 kilobase region on chromosome 4H, and then we used 3 main strategies to identify the genes that are required for resistance. So first of all, there was a transcriptomic study with a wide variety of barley accessions. And out of all the genes in this locus, there are only 2 that have an expression polymorphism. These are XO70FX12 and PER1. And so the expression of these 2 genes predicts that barley genotype will be resistant. Additionally, we used a forward genetic screen using loss of function mutant screening. This is a TM population, and this is made possible by collaborators at the University Bologna in the Silvio Salvi group. And this population consists of mutagenized Morix lines. Morix happens to have RPS8 in isolation as far as wheat stripe rust resistance loci. So we began screening this population for loss of function mutants and identified loss of function mutants with causal mutations either in PER1 or XO70FX12. So because of these mutations, they lost that resistance. Lastly, we performed a transgenic complementation test, and this figure shows the infection score from that test. We use a 0 to 4 infection scale, where 0 is resistant, and 4 means that the leaf is completely covered in pustules, so it's susceptible. So here we have a resistant line as a control, and then we have 2 susceptible lines. S by GP DH47 is also transformable, so that's the background for these transgenics. When this susceptible line is transformed with XO70FX12 or PER1 individually, it does not gain resistance. There's only resistance when both XO70FX12 and PER1 are both being expressed. So in conclusion, these 3 layers of data support that PER1 and XO70FX12 are both required and sufficient for RPS8-mediated resistance against wheat stripe rust. After defining that they're required, the next big question is, what are their molecular functions? And we used a phylogenetic approach to begin making predictions as to the molecular functions of PER1 and XO70FX12. To begin, I'll talk about PER1. PER1 belongs to the LRR12 subfamily. LRR stands for leucine-rich repeat, and this is due to the ectodomain of these receptor kinases, which has leucine-rich repeat motifs, and is responsible for binding to ligands in the extracellular space. This subfamily is quite famous. Many different receptor kinases involved in plant immunity have been studied. These include FLS2 in most higher plants, EFR in brassicas, and XA21 in rice. So each of these 3 receptor kinases perceive a peptide from a bacterial species and then transduce immune signaling inside of the cell. PER1 has a very similar structure to all of these receptor kinases in that it has the extracellular LRR domain, transmembrane domain, and intercellular kinase domain. So how closely related is PER1 to these other receptor kinases? It turns out that PER1 is the ortholog of XA21. Here we have the genetic tree of LRR12 proteins in grass species. And you can see the organization into clades, supported by bootstrap support of 80%, indicated by these red dots. And then on the right, we see an expanded version of the XA21 PER1 clade. And we see that XA21 and PER1 are in the same clade, supported by bootstrap support of 95%. And this shows that these two proteins are orthologous. Based on the function of these related receptor kinases, we predict that PER1 has a very similar function responding to a ligand from Puccinia striformis formispecialis tritici, or wheat stripe rust in the extracellular space, and passing along that signal. I won't have time to talk about it today, but we do have evidence to suggest that the kinase domain of PER1 has functionally diverged from XA21. Additionally, we know that unlike these other receptor kinases, PER1 requires exo 70FX12. So I'll next be talking about the phylogenetic tree of exo 70FX12, which becomes quite interesting. First of all, what do exo 70 proteins do? They're canonically involved in the exocyst complex. The exocyst is involved in exocytosis in the cell, so it is integral to tethering these post-Golgi vesicles to the plasma membrane prior to sterol-mediated fusion. And the exocyst is evolutionarily conserved. So it was discovered in yeast, and it is in fungi, animals, and plants. However, exo 70s in plants are very interesting, and so I'll be going through 5 different facts of exo 70s in plants that are pertinent to this project. First of all, exo 70s in plants are highly diverse. In all other species other than plants, there's only one copy of an exo 70 in the genome, and this is the case for yeast, animals, and fungi. However, there are upwards of 20 copies of exo 70s in each plant species, and they organize into at least 9 different clades. Additionally, we know that exo 70B proteins are involved in receptor kinase immunity. We know this from a paper that was published in 2020 by Wang et al. And it showed that in Arabidopsis, exo 70B1 and B2 interact directly with the receptor kinase FLS2 and are required for accumulation of FLS2 and other receptor kinases. So that was the exo 70B clade. When we take a look at the exo 70FX clade, things get quite interesting. This clade is only found in grass species and is highly expanded. Here's a phylogenetic tree of exo 70s in grass species, and you'll immediately notice just how expanded the FX clade is. Then, looking at the different clades, we see very short branch lengths, and you can see this in example with the exo 70B, as well as all other clades, other than exo 70FX, and we have very long branch lengths indicating highly sequence divergent proteins. Lastly, another unusual characteristic about exo 70FX members is they lack critical domains that are involved in binding that exo 70 protein within the exocyst. For example, the core X domain in exo 70 is integral to its partnership with another protein in the complex, and we find that that domain is often lost in exo 70FX members, and that includes exo 70FX12. So based on this genetic evidence, we have reason to predict that there is that exo 70FX members are undergoing neofunctionalization. And so we've proposed two main different hypotheses for the mechanism of exo 70FX12 and PER1 based on this phylogenetic work. In hypothesis 1, we predict that exo 70FX12 is required for accumulation of PER1 at the plasma membrane, and this could either be in an exocyst-dependent or exocyst-independent manner. The second main hypothesis is that exo 70FX12 is involved in the signal transduction of PER1, and the ultimate alternative hypothesis is that it's involved in some other downstream pathway related to immunity. So one of the critical first questions to answer is whether or not exo 70FX12 and PER1 directly interact. And we approach this initially with a yeast-2 hybrid assay. So if these proteins interact, you'll see a colony growing on the second medium. Beneath the controls, I've highlighted these key tests, and we test interactions both with exo 70FX12 and the full-length PER1, and exo 70FX12 with just the intracellular truncation of PER1. And in both cases, we found that there is no interaction that we can identify. So the next steps will be confirming this lack of interaction with in vivo tests with co-immunoprecipitation, And then we'll be studying whether or not Exo70FX12 interacts with other members of the exocyst with similar methods. So to conclude, I'd like to come back to the hypotheses. And we predict that PER1 and Exo70FX12 are providing resistance through a novel mechanism. While we cannot yet disregard any of these hypotheses, we do favor hypotheses in which there is no direct interaction between PER1 and Exo70FX12 based on these yeast-2 hybrid results. And this is in contrast to what we know about the Exo70B clade members, in which they provide a role in receptor kinase signaling by directly interacting with the receptor kinase. So from this project so far, we've discovered a function for the Exo70FX clade member, and we predict that more members have specialized roles in plant immunity. Additionally, PER1 requires this grass-specific Exo70, and it looks like it has functionally diverged from XA21, its ortholog, based on this dependency and based on interesting information about the kinase domain. In conclusion, I'd like to thank all of the Moscow group, especially Matt for all of his mentorship and teaching, Diana, Inma, and Fawn for all of their help in the lab and greenhouse. So with that, thank you so much for your attention, and I'll take any questions.
Awesome talk. Have you guys considered doing yeast-2 hybrid library screens, both with the extracellular portion of your LRR and with Exo70FX, just to see if there could be an intermediate interactor that possibly does? And granted, that would be with the kinase domain if they interacted together, but.
Yeah, that's definitely a good thought. And we predict that the Exo70FX12 is interacting in the intracellular, with the intracellular components of the receptor kinase. So that would be one strategy, in addition to pull downs, which we're also going to work on.
So you mentioned that you have evidence that this exoprotein is not necessary, for example, for the XA21 factor in rice. What is the evidence for that? Because it could be that maybe it is necessary, but it's maybe localized in a different place, or all the lines that carry the XA21 gene happen to have the Exo70 protein.
Yes, thank you. That's a great question. XA21 has been transformed into several different species, including dicot species, and has been proven to provide resistance. So based on that, we know that it doesn't require FX members because those dicots wouldn't have an Exo70FX member in them. So that's the main evidence of that. Thank you.
If I remember correctly, Exo70s are also responsible for vesicle transportation from the endoplasmatic reticulum to the cell walls. So did you ever look into cell wall depositions after infection of fungal pathogens or something like that?
No, we haven't looked into that, but that would be a great thing to look into with like callus depositions and the defense in that regard. Thank you.
Before I start my talk, I would like to appreciate local organization committees for giving me the opportunity for video representations. It's really a pity that I'm not able to attend the RIGA for this wonderful events because of problems in international traveling. Today, I would like to show some works in biomass resistance in barley and wheat. So my talk is convergent mechanism of host susceptibility factors assisting the bioviruses infections in barley and in wheat. As you can see here, there is a barley field pictures where the barley plants were infected by the barley yellow mosaic virus in Germany. The infected plants show yellow discoloration and produce less thinners and fewer seeds, and finally result in a yield reduction. In my talk, I would like to include two contents. First, I will briefly introduce the soil-borne bioviruses that infect cereal crops, and then focus on the genetic resistance against the bioviruses in barley and in wheat. Plant viruses are constantly destabilize the yield productions in cereal crops For instance, in 1950s, the occurrence of barley yellow dwarf disease caused a severe yield reduction in barley and in wheat. And in 1970s to 1990s, the barley yellow mosaic virus disease has caused severe problems in winter barley productions in Europe and East Asia. The yield reduction caused by plant viruses infection have been repeatedly reported in several cereal crops. Plant viruses are obligate microorganisms and survival depending on the host species. For most of plant viruses, their transmission are depending on the insects or the fungi. Their infection relies on the insects that beat the upper ground tissues or the fungi in the soil that penetrate the plant roots. Plant viruses in generally with simple genomes that encode for only few proteins, and they have to hijack the host encoded components to perform the life cycles. However, they modified the host factors in the plants are able to result in the recessive resistance. In cereal crops, there are 10 host factor genes have been cloned. And for us, we are specifically interested in these genes that interact with the soil-borne viruses. So the viruses transmitted by the soil-borne fungi Polymyxa graminis are serious problems in cereal crops such as wheat and barley. So this intermediate fungi vectors is able to transmit 11 identified viruses from two genus, Bimovirus and Flovirus. Especially for the Bimovirus, the barley yellow mosaic virus and barley mound mosaic virus are specifically to infect the barley, while the barley spider strip mosaic virus and wheat yellow mosaic virus only infected wheat. The Bimovirus disease of barley was known to be a problem in winter barley production in Europe and in East Asia. So this disease has been reported in over 20 countries. In 2014, this disease was reported in Iran, that suggests this disease is still spreading. The Bimovirus disease of wheat have been found in several countries from continents. The wheat yellow mosaic virus is occurring in China, Japan, and India, while the wheat spindle streak mosaic virus was found in North America, Europe, and also the Zimbabwe's in Africa. In the field conditions, the Bimoviruses are present in the resisting spots, which protected for the virus particle, and also the vectors can germinate and penetrate the host roots under the favorable conditions. So the virus can replicate in the host plants and translocate the virus components from the root to the upper ground leaves. That's why we can see the disease symptoms on the young leaves mostly in the early springs. Several cultivation approaches are able to reduce the disease severities, such as fungicide treatment, crop rotation, and later sowing. However, breeding for virus-resistant varieties is certainly promising in disease control. Although viruses are simple with small genomes, cloning of disease-resistant genes against viruses are difficult. Up to date, over 60 resistant genes have been isolated to fungi and bacteria, while only 2 resistant genes against viruses are identified. Therefore, identification of virus-resistant genes becomes of interest. In barley, there are 22 resistant genes at 16 genetic loci were reported, while in wheat, there are 12 resistant genes at 7 genetic loci were reported. It's very surprising that in barley, most of resistant genes are inherited in a recessive manner, so this type of resistance is usually associated with the modification of host factor genes. However, in wheat, none of resistant genes are recessive, and all resistant genes are speculated to respond the active recognition of the virus-initiated components. The first virus-resistant gene in barley was cloned by Niels in 2005. This gene encodes for the eukaryotic translation initiation factor 4E, and its anemic amino acid variants RAN4 and RAN5 confers race-specific resistance, and both alleles have been widely used in winter barley breeding in Europe and East Asia. However, both resistant alleles have been overcome by appearance of new virus isolates. I cloned the second barley virus-resistant gene RAM11. So when I was a PhD student in Niels' group, so this work also in collaboration with Frank Alden's group. So this gene encodes for protein disulfide isomerized like 5-1, and loss of the wild-type allele were resistant to the virus infections. We also demonstrate so the former resistant genes RAM1 and RAM11 are allelic, and both genes are renamed RAM111. EF4E was considered as a host-typing gene since we didn't observe the loss of functional alleles in barley later populations. However, a recent studies from Johan Kuhlmann's group observed this gene is dispensable for barley itself. Locking out of this gene results in barley mild mosaic virus resistance and also produced fewer grains per plant. That means locking out of barley EF4E would result a yield penalty. In my group, we conducted a genome editing for barley PDF5-1 genes and identified 2 independent transgenic events. We obtained 1 lines with 3 base pair deletions that encodes for 1 base pair amino acid deletion. So this lines is also able to suppress virus accumulation and finally result improved virus resistance. So this allele is different with the resistance that caused by loss of functional genes PDF5-1. We also conducted agronomic performance in a garden experiment, but we didn't observe yield penalties. So that means this gene is a desirable target to edit for virus resistance in commercial barley varieties. As we know, the bamoviruses in barley and wheat are grouped together, and this virus with a similar genomic organization. We're simply asking whether the host factor genes identified in barley are functional similars in wheat. Barley is a diploid, while the bread wheat is a hexaploid. We speculate the polyploidization in hexaploid wheat has resulted in functional redundant genes that compensate for the genetic deficiency in any single gene, therefore blocking the identification of a recessive resistance. In order to test the hypothesis, we conducted the CRISPR-Cas9 guided genome editings of the weight PDFF-1, which is the ortholog genes of barley PDFF-1 genes. So follow a crossing and a marker-assisted selection process, we identified 4 independent triple mutants, which have edits on weight A, B, and D genomes, and also we identified each single and double mutant. Under greenhouse conditions, we tested these triple mutants by mechanical inoculation. As you can see here, all the triple mutants are without yellow discoloration symptoms at the seedling stage. We also quantified the virus accumulation by RT-PCR and was quantitative PCR. We are not able to detect the virus fragments. It's clear that the triple mutants are resistant against white yellow mosaic virus. We also tested the single, double, and triple mutants. As you can see here, so the triple mutant is resistant, while other mutants are all infected with yellow discoloration symptoms. This result demonstrates that the weight PDFF-1 gene is a host factor gene to white yellow mosaic virus, and each of 3 copies can compensate for the genetic deficiency in any single gene. We also analyzed the agronomic performance in a garden experiment without virus infections. As you can see here, there are no difference at 9 agronomic trees, and that demonstrates PDFF-1 gene is a good target for genome editing for white yellow mosaic virus disease in commercial wheat cultivars. We applied the same principle to test the second host factor genes with EF4E. So the disease symptoms pictures were different with the former pictures, as these photos were taken using the translucent plate as a background. So the triple mutant resistance is without the disease symptoms, while the other mutants and the control lines are all show yellow discolorations. So it's obvious that the EF4E genes is a host factor to white yellow mosaic virus. The plants after virus test using a seedling assay were transplanted under normal glasshouse conditions. So the virus-infected plants are small and show less thinners and delayed growth. Only the triple mutants are resistant and grow normally as the uninfected mock plant. In a garden experiment, so the triple edited EF4E mutant plant is poor compared to other plants, also show delayed maturities. That means this gene is not as good as the PDFF-1 to edit for virus resistance in wheat. In summary, our results demonstrate PDFF-1 and EF4E are host factor genes for 3 soil-borne bioviruses, and both genes are dispensable for wheat and barley themselves. A common mechanism deployed by barley yellow mosaic virus and barley mild mosaic virus in barley and wheat yellow mosaic virus in wheat is present. Cloning the host factor genes in barley and deciphering their functional mechanisms are of interest in barley and beyond. In my last slide, I would like to appreciate, especially for Jing Hongkan, Yu Cai, and Chun Yuancheng, who are involved in the genome editing, Dr. Zhen Tianhe for collaboration in field trials. Some slides in my presentation are the work when I was a student working with Niels, Andreas, and Frank Ordens. Thanks for them for nice collaboration. Thanks for the funding resources. I'm so sorry here I'm not able to respond to your questions. However, I'm very happy to answer your questions by email. Finally, I would like to appreciate again to local organization committees for your efforts to organize the symposiums, which I'm pretty sure will become a successful, wonderful event in global Bali community.